in Andhra Pradesh.
You are live.
Not only pharmacy, we mean different streams. The quality education is given to Andhra Pradesh.
అక్కడ పోతున్నా వస్తున్నా.
Thank you for giving this opportunity. I am very much thankful to my guru, teacher, Professor Reddy sir. I always really take the lessons from sir. My student days. I studied in the College of Pharmaceutical Science in Vijayawada. I know really the star is here. I can say like
అదే OBS లోకి వెళ్ళి కరెక్ట్ చేయండి.
star will teach like chemistry, sometimes will teach like engineering, sometimes pharmacology. So many subjects we learn from that. Prefer to do something oral drug delivery system. The oral drug delivery system, almost 80% of the drugs we are using by oral route, most acceptable route. Then at the same time, based on my background where previously worked, I am not getting that much opportunity to do the research. Whichever is available in my college, that I thought to work. So these are some of the reasons I was interested in work on oral drug delivery system. The question is here the research gap.
61 members వచ్చారు.
As per my knowledge, the research is nothing but here. You need to find the problem to be the solution. The problem is nothing but here. What is the gap? The existing work, the research. You need to find a gap, research gap. The oral, the control delivery system, generally the identification of the research gap, I try to always suggest three important things. One is which area is having less contribution or less information, that is take it as a research gap. The second thing is the limitations. Of course, every area is having some limitations. The advantages are there, at the same time limitations also there, disadvantages. That is the research gap. The third thing is controversy. In research, different controversial statements will be there. It will go like debate. I found the three in this image independent controlled and delivery system.
Sorry for the interruption.
Please?
Sir, your slides are not visible, sir.
Slides are not visible.
Yes, sir. I think your screen is not in sharing mode, sir.
Now it's okay?
Yes, sir.
Yes, sir.
Yes, now it is okay, sir.
Thank you. Yeah. I think voice is clear now, audible. Okay?
Yes, sir.
Thank you, madam. So these three important points I will always consider and suggest research scholars who are under me, they are doing the research. Found a research gap. Research gap, I think, is very, very important. Regarding the pH independent drug delivery system,
ఇప్పుడు తీసుకోవట్లేదు ఫస్ట్ లో అది ఇంకా
I saw many books.
తీసుకున్నట్టున్నమాట.
I read many books like ANDHRA, VAS, CHIN, many international books also. I studied, I read it, many articles, many books. So very little
అది
information
ఇంకా ప్లే చేయలేదు.
about pH independent controlled by delivery system.
ఫుల్ స్క్రీన్ వచ్చింది.
ఇందాక
Very
చేశాను.
few research articles are available. And then I thought it why very less information was there. Of course, if you go for like maybe dissolution or diffusion or floating drug delivery system or osmotic or annexation, plenty information is available. But this pH controlled drug delivery system will not get that much information. Then I thought, why? What happened? Why people are not focusing on this area? Then I started to study. Then I found it. The N.K. Jain, he expressed like I saw some of the articles also. Then you know that controlled drug delivery system advantages and disadvantages. I found it. pH dependent soluble drugs are not suitable for controlled drug delivery system. Then again I studied it. Then I discussed many eminent professors like Ramamurthy Garu, Andhra University, and Nageshwaran Garu, Patna University, or my the guide, that Gopal Krishnamurthy. I interacted with different Dr. Reddy's lab or with me. Then when we are discussing, there is a controversy. I presented even Rajasthan, like Bharat, this project proposal was I presented. There was also
ఇది మన వాయిస్
the controversial sectors of things.
కంప్లీట్ గా రికార్డ్ అవుతుంది. ఆ
Then I
యూట్యూబ్
started
మనం మాట్లాడి
to do something
ఇప్పుడు కూడా
in this area. I need to learn, I need to do something. I need to learn something from this area. I need to contribute something. Then I tried to do something. You know that
మైక్ మ్యూట్ చేయండి.
gastric, the oral route, the pH is one of the important factor. It influences drug release. You know, the pH is a complex process in GAT. Need not worry about like conventional dosage form, but when we are going to design controlled drug delivery system, it's a complex. You see here like stomach 1 to 3,
దీంట్లో
intestine 6 to 8, large intestine 5 to 7.
మన వాయిస్
Like the
దీంట్లో మ్యూట్ చేశారు.
the pH
మ్యూట్ చేస్తే అది
is a complex process.
యాక్సెప్ట్ చేయట్లేదు.
సిస్టంలో మ్యూట్ చేయండి. సిస్టమ్
That will be keep your mind. The next thing is
ఇది
solubility. And if you always one word, Sharjil book, you mentioned one word.
మన టీమ్స్
I like that sentence. The solubility is one of the important parameter for viability in oral drug delivery system. Drug available in solution form at the site of absorption. That is very, very important. what type of dosage form you will take? Maybe tablet, maybe like spear or whatever it may be. But it must be available in solution form at the site of absorption. Then only the absorption is possible. Then drug will be entering the systemic circulation. Then bioavailability. Otherwise, impossible. So that's why here the solubility of the drug, it plays a major role for better or pharmacological activity in oral growth. See here, I can give one example of the drug. It's really surprising. The diazepam. See, the pH 1.2, 30 mg per ml, then 3.2, 23. Then you will see 6.8, 0.046 mg per ml. This is like basic medium, 6.8 phosphate buffer. This is for acidic medium. See the solubility, 0.046. Drug is not at all soluble here. Here it is completely solubilized, pH 1.2. Then definitely we can say this drug is called as pH dependent soluble. Then what the solution? How we can get a solution to the type of drugs? Another drug is also same like ondansetron. This generally we are using for chemotherapy, nausea, vomiting, induced nausea, vomiting. The frequent drug must be required during chemotherapy, but the oral is very difficult for control prolonged. That's why the conventional dosage forms are available in the market. No doubt it, the pH independent is the superior to do something to make a formulation for better industrial application. If you know, like many reasons for that behind it. To take 8 patients of the decreased acidity in the stomach, therefore significant effect on the oral bioavailability of pH dependent soluble basic compounds. At the same time, you can see like old people, hypochlorhydria, gastric acid secretion leading to gastric pH value more than 5. The problem is here, the pH will be increased. Mostly the problem is with weekly basic drugs. As it is not that much a problem. pH partition hypothesis theory, irrespective of either stomach or intestine, whatever it may be, the major absorption site is intestine. Problem of again here, the basic drugs, those drugs are like dependent soluble drugs, are the solubility is one of the major drawback of the intestine, like basic pH. That we can't get it the better pharmacological action. Already we know that. Any drug, it must be available in solution form at the site of absorption. Then only possible for absorption. That is a general basic principle of drug absorption. So these are the drug drawbacks. Inter-subject variability. You know that inter-subject variability is one of the major drawback, because the HCl secretion or the pH will be varied, depends on disease state also. Precipitation of the basic drug it will be precipitate. The complete solubility is questionable again here on the oral route. Poor in vitro in vivo correlation. You know, it is one of the drawbacks of oral controlled drug delivery system. Subtherapeutic doses will not get it therapeutic action. It leads to poor bioavailability, the variation in disease conditions. Among all this, generally we know that nowadays doctors will prescribe all the time along with the drug. They will give some like antacid like Zintac or Amphetamine. We know the pH, the variation. So these are the drugs for mostly the ideal drugs for pH independent control drug system. We need to design for better the therapeutic activity. pH dependent soluble drugs like more readily basic drugs. So absorption inculcated to the relative narrow window. Those drugs are the precipitation GIT, the potent drugs like onacetran. Drugs are required for prolonged therapy. These are all hello.
Sir, your slides are not.
Moving or not?
No, sir.
Actually, previously moved now.
Sir, press the glass pot watch at the bottom. You have the bottom. Can you see the bottom of the page?
Yeah.
Next to percentage, you please click that one.
Now it's okay.
Yeah. Click that one. The third icon. At the bottom, sir. At the bottom of the slide. Can you see a
Right.
jar thing? Click that one, sir.
It's okay now?
No, sir.
Sorry.
Next to the percentage, can you see one icon over there?
Where?
At the bottom, next to percentage, 64% is shown, no? At the bottom of the slide.
Bottom of this slide.
Percentage,
Anil?
next to percentage.
Sir, hello.
Yeah, please.
సార్ ఏం లేదు, మీకు రైట్ సైడ్ టాప్ లో షేర్ అని ఉంటది. దానికన్నా ముందు మీరు ఏం చేస్తారంటే పవర్ పాయింట్ ప్రెజెంటేషన్ మోడ్ లో పెట్టుకోండి. షేర్ అని పెట్టేసి అప్పుడు సిస్టమ్ అంతా సిస్టమ్స్ కదా స్క్రీన్ షేరింగ్ కొట్టేసిన తర్వాత అప్పుడు ప్రెజెంటేషన్ మోడ్ లో ఉన్నది సెలెక్ట్ చేయండి, అయిపోతుంది ఈజీ. ఓకేనా సార్? ఫస్ట్ ప్రెజెంటేషన్ మోడ్ లో పెట్టుకోండి సార్, అది పీపీటీ.
Now it's okay.
Then share.
Yes, sir. Now it is okay, sir.
Yeah, thank you. Some the benefits of independent formulations. No doubt it. The inter-subject variability is also it will be solved. Drug stability is also it will be increased. The individual correlation is also it will be enhanced. Less toxic. So these are the list of some of the drug examples. As just I was mentioned few examples. Many drugs are available till market. Due to the dependent solubility, they are not designed to control drug delivery system. That's one of the drawbacks. I just given a few examples, but I have many drugs. There are some patentable already. These drugs are having some patent pH independent control drug delivery system. The Reddy Biopharma, they tried to design. They took the patent independent drug delivery system. So this is also on Actalis pharmaceutical technology. They designed like the DQ cap technology. So this is like sustained release. The layer will be there. The core, the organic acid will be there. The inert particle core will be there. Like one is organic acids, another one is alkyl. Maybe both drugs, like SD drugs, are basically both will be designed. But when I study this technology, it's very costly, at the same time complex process. Then I tried in my laboratory different strategies. One is like it's a common strategy, pH modulating agents, means by using organic acids like citric acid, tartaric acid, fumaric acid, succinic acid, even many acids also available. The strategy is combination of anionic and non-ionic polymer. Here the non-ionic is HPMC and it is sodium alginate. The polymer precipitation inhibitors by using some PPR, the polymer precipitating inhibitors. Okay, like polyethylene glycols, polyethylene mud, and non-polymeric precipitating inhibitors also by using like cyclodextrins. So these are some strategies. Generally preferable is executing to solve the problem of pH dependent solubility. It will be fluid. You know that the low density drug delivery systems are suitable for weakly basic drugs. High density systems are suitable for weakly acid drugs. It's a general principle we are following. Out of that, gastroretentive drug delivery system.
செக் பண்ண மாட்டாங்களா
Long period of the drug release is a question mark. That's a different role. I can say like in vitro correlation is very poor because we know that any the drawbacks are there, like water is required and the pH is also one of the variation. Like say osmotic drug delivery system is also is the solution to design like pH dependent. But again here, the many parameters we need to optimize. Many factors we need to consider here again. Osmotic pressure, thickness of the membrane orifice. So to make it, it's very tough. The many factors, it's very difficult to prolong the constant release entire the GIT. The next thing is pH modifiers. Most of the literature is available, but still it's having many drawbacks. These pH modifiers. So you know that it's simply the concept is here alteration of micro-environmental pH. It should maintain the pH modifier. It alters the micro-environmental pH, the surface of the tablet. We know the surface pH is very important than bulk pH. The basic principle what we learn in the dissolution concepts, theories also. So here, based on that theory, what we found, what we hear that these pH, like the surface pH, is modified by using pH modifiers. But one of the drawback is here. What I found it, I tried many like organic salts. The selection of appropriate modifier is very important. That's one of the drawback. At the same time, drug, the pH, to maintain the surface pH for prolonged time is also difficult. By using anthric polymers, we know that anthric polymer is acting as a like acidifying agent. Still, the coating of the polymer is again so the drawbacks we are facing for constant drug release is long. The combination of non-ionic and ionic polymer is also another strategy. Both like sodium alginate and HPMC. The sodium oxalate is an anionic polymer, HPM is an anionic polymer. Both we are mixing. So what happened here? When we found it, it should maintain when they reach the like small intestine. The sodium oxalate, it will be swallowed, then drug it will be released. But the thing is, problem is here. The problem of in the lower pH is lamination. It like cracked. We found the when it has like burst release of the dosage form. The drug was released immediately. That we found it this approach. pH dependent saturation is another the technique. Means here the super saturation. So by using like nucleation inhibitors with the combination of pH modifiers, we tried it, but the drug solubility is not enhanced that much. See here how the formulations pH 1.2, pH 5.5. At the end of formulas, so we took the different concentration of the polymer. The pH 1.2 100% drug release, but in 5.5 the drug release will be less than 10%. Almost all the formulations. When we are increasing the concentration of HPMC, what happens? The drug is controlled, but the large variation between pH 1.2 to 5.5. This is generally from the optimized formulation. Some graphs by using different approaches. Another one important challenge pH independent formations. I tried many tribes, different strategies, but the drug release is not up to the mark. All the time it's a failure. Then I thought it. I studied in the different authors, different differences. What I suggest here, whenever we are going to formulate any dosage form like independent drug release, first of all we need to develop dissolution method, because drug where it is solubilized, which pH, at the same time how much volume, method development must be required. Otherwise, we will not get it. What the results what we are getting. Suppose you see here, the 900 ml, the buffer medium, 5.5. See, the drug is precipitated. Same 6.8 phosphate buffer here. Drug is not at all solubilized. Same pH 5.5 phosphate buffer. Drug was not precipitated. The volume is 500 ml. Drug was solubilized. So this is like I was make it like dosage form. I tried. Then finally I found it. The dissolution method. The best one is the 500 ml is. I choose the 500 ml the volume, and optimize like 100 RPM also. Like these are the like dissolution data, the different pH mediums, different volumes. So finally I conclude this is the dissolution development method with 500 ml, 100 RPM. The pedal method is the best. These are the some the formulations. I tried different combinations, but not work out. So this is one of the technique. Core and coat. Composition of compression coated technique is one of the best technique for pH independent controlled system to prolong, maintain pH at the surface of the pH. So that's why first of all I prepare like core part. The core part and core part is also will be there to control the regularities. Then I compress by a tablet compression machine. Then we saw the best results here. See another the big challenge. See, this is a matrix tablet. What happened? I think the picture maybe you found it. Because we know the salts are, it can easily solubilize, the moisturize. So then the tablet, see, it is like wetted. Then another one is the compress coated only. You see here the capping. So we prepared the granules during the compression coating. This is matrix tablet only, like in form like this. The compression coating we saw here, the capping. Then finally we got it excellent the tablet, the compression coated tablet with pellets. I'm really surprising. I tried many trials by salts, but I thought it how to control these problems. Then I got it one idea. This salt we will make it like pellet. It will be not that much. It will be solubilized. So that's why this Woman Pharma is one of the company was there. I approached them. They are preparing like pellets, organic salts in pellet form. This is like 2019. I got it. Okay, from that company, different organic salts. The advantage of these the pellets, the salt in a pellet form. One is the major drawbacks of these organic salts. It can be solubilized very fast. It diffuse from the tablet. Then what happens for prolonged to maintain the surface pH is very difficult. But in a pellet form, we found it the surface pH is prolonged until the drug release. That's the one thing. The second thing is the drug which found it the device dosage form. The stability was improved. It was not exposed until when the dissolution. We found it, the stability is excellent. Reports we got. So here are some reports. So these are the different matrix. This is like a matrix tablet, tartaric acid pellet. We found it the same organic salt in a pellet form. The drug release will be high. Not only that, the stability was improved. So we studied different pH like pH 1.2, pH 5.5. The drug release compared. Some stability studies after my formulation. And potent drug like onacetrol also we made for pH independent control system in our laboratory. It's excellent results. Then the future, the scope, there is a need of extensive research on pH-dependent extended release formulations for various categories of the drugs. We need to study because many the drugs are available in market, the dependent solubility drugs, which are not available in controlled drug delivery system. There is necessity to establish the scale-up techniques for industrial applicability. The scale-up techniques are not available. We need to develop some industrial applicability. That was one of the future scope. The other thing is multifunctional polymers. A polymer is having a multifunction. It works like the pH in the pH independent polymer, control release, and the polymer it should maintain acidifying character. Like such a type of polymers we will make in future. Definitely, it will be useful for better drug release to design pH independent control drug delivery system, multifunctional polymers. There is necessity for establishment of in vitro model in vivo performance. Still, there is a confusion how to make up with in vitro in vivo for this pH independent control drug delivery system. That need to develop. These are some recommendations. In future, anyone is going to do that. The use of combination of pH modulating agents was best. I found the previous research. The most of the people they use just only like single agent. Instead of that, like to use the multi pH modulating agent, the best drug release is saw. Next, the pellet form is excellent for stability and to maintain the better pH. Next, the tartaric acid, turmeric acid is excellent pH modulating agents than citric acid, succinic acid. The thing is the compression coated tablets they produce better pH or prolong the drug release. The next one is multi-component compilation techniques. It is also a good technique. First, initially we need to enhance the solubility of the drug by using beta-cyclodextrins. Then we will control the drug release in a proper way. The dissolution method development is required. Multifunctionals is required. So finally, what I am doing here, concluding, why these many techniques? The thing is here, no approach is universal applicable to all pH dependent soluble drugs. The method, we can't say, okay, this method is best for this drug. We found it like that. Each drug is having the method is suitable. It depends on. That's why we need to trials are required many. There is need of research work. The selection of the suitable method is not that much easy. It depends on drug to drug. It will be varied. These are some differences. The articles I published on this pH independent drug delivery system. So I conclude here with my lecture. So thank you. now I hand over to organizing team.
Thank you, sir. It's a very informative, engaging session, sir. Thank you for spending a valuable time, sir. Participants, if you have any questions regarding this topic. Participants, if you have any questions. I think there is no questions.
Yeah.
Thank you, sir. We greatly appreciate you, sir. The time and effort you invested to share the expertise with us. Thank you, sir. Very informative session, sir.
Thank you for the kind attention for all. Bye.
We can conclude it, sir. Thank you all for active participation. We'll see you tomorrow with another good session, engaging session. Thank you.